This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
So while you do that, I'll just say a few words. So Jess Taubert is an ARC Future Fellow at the University of Queensland. She has a range of accolades and a pretty impressive track record on face processing, both in chimpanzees, macaques,
non-human primates, animals, chickens, dogs,
exactly.
You name it. Exactly.
So that's pretty good. And Jess has been traveling around the world doing this research for
a long time. Too long, yes.
So Jess has been at the NIH with Leslie and Yolanda, which I'm sure rings a bell to most of us. And she's been in Belgium, also in the US in another.
Emory.
What was it?
Also at Emory.
Emory
University.
And Sydney, of course.
That's where I did my PhD.
And now at UQ. So all over the world.
Different places, different faces.
Exactly. So with that, I leave you to take it away.
Okay. So thank you very much for the introduction, but also the invitation to be here. What a fantastic experience, and with a minor setback with Qantas cancelling all my flights, everything has been going extraordinarily well. So thank you for everything and organising everything, Janet. It's been great. I did design this talk to be a little bit of an overview, so as a consequence it's going to be a little light on methodological detail, and it's also still a little long, so when I was going through it last night I was pulling out slides, so there's going to be some very obvious kind of discontinuities in some of this. But the good news is I'm around all day and all day tomorrow, so anything that I sort of really quickly gallop through or don't explain well, there will be time to explain all of those details later. So without further ado, our current model of primate vision assigns object recognition to the ventral visual pathway and then divides the ventral visual pathway up into several processing stages, primarily feed-forward. I think anyone that comes from Leslie Ungalaida's group is kind of obliged to start any talk with this particular image. But of course, this isn't the only way that we depict visual processing. This is another way. And no matter how it's visualized, the general idea is that we see an object, its image is projected onto the retina, converted into electrical signal that is fed into the early visual cortex, where low-level visual attributes are extracted. There's then some pooling at various spatial scales. The next stage involves comparing the incoming visual input to stored representations of previously seen shapes and objects. Somewhere in there is the distinction between high-level and low-level visual processing. And as a result of all of this processing and these comparisons, the brain is ultimately able to classify and label an object. Now, the modern metaphor for this hierarchical processing pipeline is, of course, the convolutional deep neural net. And these days, in particular these days, DNNs are extremely powerful and sort of freakishly accurate. For example, you can now feed a completely novel test image like this one to a pre-trained DNN, essentially off the shelf, and it will likely tell you that there is a high probability that this image contains a dog. And depending on the architecture of the DNN that you're using and how it's been trained, it may also flag the tiled bathroom and the clothing on the floor. So this is a lot of information. However, as impressive as our technology is, I want to argue today that we as humans can read out so much more information than any given machine that we currently have available. For example, despite not witnessing the previous events that happened before this image was taken, we know exactly who was responsible for making this mess. So the processes that underlie our perception, our phenomenal experience of the world, go beyond the putative classification of visual features and objects. And in my lab, we are specifically interested in the detailed information that we can read out from visual scenes by looking at the faces of the people around us. Now, interestingly, primates are equipped with specialized cortical regions for processing faces. These regions are sort of marked on this toy brain here with little circles. These functionally defined face-selective regions are tightly interconnected, yet each is thought to play a distinct role in face perception. And we see a strikingly similar face-selective network in the macaque brain. This observation not only adds weight to the general feeling that face perception is very important to social primates, it's also allowed us to seek descriptions of face processing at multiple levels of neural organization, so from single cells to neuronal populations, all the way through to whole brain circuits. Not to mention that having a nice validated non-human primate model is a valuable tool for linking brain activity to behavioral outcomes using targeted causal methods. Yet, despite all these blessings and advantages, we do not yet understand how faces are processed, and the scientific community is really super uber divided and disagrees about fundamental aspects of this network, including the degree to which its definition is epiphenomenal. So moving forward from here, the good news is that we now have really powerful instruments and really great methods for probing the neural representations underlying sensory experiences. The bad news is that our behavioral methods are markedly less sophisticated. For example, the status quo in experiments designed to tap into high-level representations tend to take this particular form. So we force our subjects, whether it's a human or a non-human primate, to fixate at the center of a screen, and we do this one way or another. We either tell them to fixate on a central fixation cross, or we trick them into it by giving them some little task that they're doing at fixation. But either way, they're now tightly fixated on the center of the screen. And then we flash stimuli behind the attended fixation point while we record brain activity using a variety of different methods. This is it. This is the state of the art. This is where the field is. And without realizing it, we inherited this experimental format from psychophysicists who placed an early emphasis and a strong emphasis on precise and controlled stimulation of the retina. This was adopted by electrophysiologists when they were initially started with sedated animals, but this protocol wasn't updated when we were then recording from animals that were awake and fixating. And then later on, human researchers adopted this same protocol when functional imaging came of age. So all of these experiments are just using the same experimental format, flashing these visual stimuli behind a central fixation point. This is just what we do. Further, because of this hierarchical model of visual processing and its kind of prevalence and influence in the field, folks like myself who have been trying to study face perception have almost desperately wanted to adhere to the same rules as the people who are studying low-level visual properties. Consequently, instead of thinking about primate psychology or our phenomenal experience of faces in the real world and sort of working backwards from there, we reduced face stimuli to eerie humanoid or morph stimuli to give ourselves absolute control over the low-level image properties in faces. And then we designed sort of rudimentary face tasks that drive participants to attend to local features because that is the only information that we allow to vary. In sum, our general approach in the field of face perception, while understandable given the collective weight of the scientific record, lacks any kind of ecological validity. And now we are dealing with a number of theoretical bottlenecks, and we're engaged in heavy debate about the neural mechanisms underlying these tightly controlled but heavily circumscribed tasks. So what I'm trying to do now is bring more experimental psychology into visual neuroscience, which basically means employing better measurements of perception and visually evoked behaviors, and using those observations to motivate neuroscientific experiments. I think I'm a little bit behind myself, but I think you get the point. Okay, to showcase this approach, I thought it would be nice to give an overview of a couple of different questions that are currently being asked in my lab. For example, what is a face? Very important question if you're going to study face perception. How do we recognize facial behaviors, or otherwise known as facial expressions? And also context dependency. As I alluded to at the beginning of the talk, I'm not going to have enough time to gallop through all the experiments that are taking place in my lab, so I'm going to do my very best here. To address these questions, I use a wide variety of different techniques, and obviously I strongly emphasize behavioral measurements, and sometimes that involves good old-fashioned button push tasks, but sometimes that also involves measuring viewing behavior, and optimally, and a lot of the times, both of those things. For identifying mechanisms and circuitry in the brain, I typically use fMRI, and for describing temporal dynamics, EEG, MEG, and single unit recordings are my poison. I've used all of these techniques across multiple species, but primarily humans and rhesus macaques, though, as we mentioned, I've worked behaviorally with a number of different primate species, dogs, chickens, anything that was willing to be tested on, basically. And today, it's my hope to sincere hope to try and illustrate the benefits of this multi-technique, multi-species approach. Okay, so the very first question, what is a face? And by extension, what is a face-selective region or a face-selective neuron? So this cartoon I actually sort of found or stumbled upon when I moved to Belgium and started to learn how to do single-cell recordings in the macaque brain. And it sort of resonated with me and stayed with me, and it haunts me in my dreams, so I thought I would make it, bring it to your attention so that it haunts you as well. There's two characters in this cartoon. One is a gentleman with a top hat, the other is a police officer. The gentleman in the top hat says, "I'm looking for my quarter, I dropped it." The police officer says, "Did you drop it here?" The guy in the top hat says, "No, I dropped it 2 blocks down the street." So the police officer asks, "Well, then why are you looking for it here?" And the guy says, "Because the light is better here." Now, like I said, this haunts me a little bit in my field of face processing because I think the underlying message here is that if we let technology dictate where we look, we limit what we can actually find. And I think this is a deep cut in the face perception literature because of our reliance on functional localization. Meaning that when we ask a question about face processing, we typically first define our regions or neurons of interest using statistics, but we tend to ignore other observations. So baked into this approach is the assumption that faces are fundamentally special, and therefore they must activate dedicated neural mechanisms. So standard localizing procedures distill face-selective mechanisms using contrast between stimuli like these, and people are now challenging the inherent circular logic here and the assumption that faces are indeed special. Further, there are a number of researcher biases in the way that we select these stimuli and the categories that they represent, which people are now calling, and quite rightly, problematic. So to break out of this funk, in my view, we need to embrace messy, naturalistic visual input and ask ourselves, in the real world, what kinds of visual stimuli trigger the behavioral signatures of face processing? Face pareidolia is the common misperception of facial structure on otherwise inanimate objects, and these are just a few of my favorite examples, but we've all seen them in the real world, and the internet's kind of crazy about this thing. Importantly, examples of face pareidolia form a class of highly variable visual stimuli that are easily recognized as faces in the real world. And by bringing them into neuroscientific experiments, we have the opportunity to try and understand how the primate brain distinguishes faces from other kinds of non-face objects without manipulating or creating stimuli in any way. So in the behavioral domain, I led the team that first discovered rhesus macaques share our sensitivity to face pareidolia using viewing preference paradigms. In these studies, what we typically do is we show macaques pairs of stimuli, and the sort of seminal finding is that macaques will preferentially orient towards real faces and illusory faces more so than matched objects with no facial features. Further, when we superimpose fixation density plots on top of the corresponding stimuli, we typically see that subjects frequently fixate on the internal features of the illusory faces, especially the eyes, the nose, and the mouth. This inverted triangle pattern is typical for humans and macaques when viewing the faces of conspecifics. So we interpreted all of this as evidence that macaques perceive illusory facial features in examples of face pareidolia. Turning quickly to studies of human behavior, my colleagues and I started out in this field asking people to tell us how face-like examples of face pareidolia were on a scale that for some reason continually goes from 0 to 10. I can't tell you why we did that, but we are continuing to do that. And this approach is nice and straightforward in a lot of ways, and it still makes a lot of sense in a lot of contexts, but it's not very satisfying as a marker of face detection and categorization because these behaviors are thought to be exceptionally fast. So our first attempt to determine whether illusory facial features confer face-like advantages in a speeded task involved the visual search paradigm. Now this is an example, I know it's small, but it'll give you the gist, of the daunting kinds of visual displays that we gave to participants. And the task here was essentially to find one image of a particular green pepper in a sea of green peppers. And the target images in this task either had or didn't have illusory facial features. The results clearly show a reaction time for targets with illusory facial features, which was the first behavioral indication that examples of face pareidolia tap into the mechanisms that detect and prioritize faces. Since then, myself and others have used a wide variety of different paradigms to look at face pareidolia behaviorally, including continuous flash suppression, rapid adaptation, and dot probe tasks. One task that 2 of my students are working on at the moment involves the fast saccadic choice task, which takes away a lot of the decisional criteria issues we have in some of the other behavioral work. These results are still being unpacked, but what I can tell you is that the advantage for illusory faces over matched objects is more pronounced when stimuli are further away from fixation than when they're closer to fixation. So all of this, again, is consistent with the idea that examples of face pareidolia are erroneously detected in the visual field and prioritized as faces. So moving on, how does the primate brain respond to face pareidolia? Well, we actually started this quest using monkey fMRI. So we performed a whole brain fMRI experiment on 6 macaques, and that is like a massive sample size in my field. That's huge. Imagine that's like 200 human subjects, something like that. We presented these macaques with both illusory faces and matched objects in a standard on-off block design task. And to be clear, these subjects were awake and were viewing these stimuli in the scanner while we were scanning them, right? So they're awake, they're not sedated. What we found using this method is evidence that face pareidolia breaks into some of the face selective regions, but not all of them. So here you can see the results of the contrast between illusory faces and matched objects for one subject, projected onto a partially inflated cortical surface. This is the lateral view of the left hemisphere. Essentially, the hot colors tell us, those reddish orange colors tell us that the lateral face patches on the edge of the superior temporal sulcus are activated by illusory faces, but there was no differential activity detected here in any of the subjects in the fundus of the superior temporal sulcus. And that's where we usually see a lot of face selective activity. Down the bottom here, we have the same subjects data, this time comparing real faces with objects. And hopefully you can see, just eyeballing these 2 hemispheres here, that there's a little bit of difference between this brain, the data down here and the data up the top. We're seeing a lot more fundus activation and even upper bank SDS activation, which is fairly typical for faces. We don't see that at all for the illusory faces. So working with several different groups, both at NIH and in Europe, I have confirmed this fMRI result a few times now with single unit recordings. So this smoothed PSTH shows the average response of 66 neurons from a fundus face selective region. The red traces the response to conspecific faces, monkey faces, and then the blue and green trace are the responses to pareidolia and to matched objects. The upshot is that these neurons clearly have a preference for faces over objects, but for some reason, for reasons we don't really understand, this face selective region has the exact same response to illusory faces and objects. There's no significant differences anywhere in the time course, which is really fascinating. Through a collaboration in Belgium, I've also been involved with single unit recordings in a human patient with an electrode implanted in the occipital face area. It's a little bit more posterior than FFA. And what we found is that these face selective neurons in OFA responded very similarly to both real and illusory faces. That's the red and blue trace in this particular figure. Obviously, the illusory faces are not activating these neurons quite as much as the real faces, but still a very similar pattern of activation across the time course and a lot more activity for illusory faces than matched objects in black. This is the exact same profile we observed when recording neurons in the lateral face patches of the macaque brain, establishing a functional homology between the ventral face regions in the human brain and the lateral face regions in the macaque brain. So another very exciting project that I worked on was led by Susan Wardle, who's a talented Australian neuroscientist working at NIH. And this project involved measuring fMRI and MEG signals in human participants. So this is a human brain. It's an inflated human brain. It's rotated so you're looking underneath it. The fMRI data superimposed on top of that cortical surface shows us that the distinction between illusory faces and matched objects is being encoded by the sort of posterior part of the fusiform face area. But more interestingly is the MEG data down the bottom there, because using a similarity analysis, Susan also uncovered that there is some initial confusion between real and illusory faces, represented here by red and dark blue dots in these multi-dimensional scaling plots. This confusion between real and illusory faces is then resolved by about 160 ms after stimulus onset. But by 260 ms, we see that the representations of illusory faces and objects are now almost entirely overlapping and intermixed. So what these MEG data describe is an interesting time course that begins with human and illusory faces eliciting similar patterns of brain activity. This time course is interesting when we consider the perception of face pareidolia is relatively stable. So face pareidolia isn't the kind of visual illusion that we can really force or that oscillates from one state to another. For example, here, you don't first see this as a face and then a brick and then a face, and you can kind of force it one way or another. Instead, what seems to happen is we see this as a brick with a face and a disturbing eye injury. So what's happening in the MEG data doesn't seem to be a process that we have access to. Oh no. Just wondering, did you see the same error warning that I did?
We just had a sound.
Okay, a naughty sound. Yeah, NIH is complaining that I'm not using the NIH network. Anyway, I'm currently working with some collaborators at the University of Queensland to examine these temporal effects and their link to perception in a little bit more detail. So this project has started with a really cool behavioral task that I'm really excited about. It's an oddball triplet task. And what we do here is we just show participants 3 stimuli and ask them to tell us which is the odd one out. We don't cue them about faces. We don't necessarily show them a human face, an illusory face, and an object in one trial. That could happen, but they're drawn at random. So maybe they see 3 human faces, maybe they see 2 objects in an illusory face. We're just trying to get an understanding of the perceptual similarity in these images without cueing the subjects about what they should or shouldn't be looking for. When we look at these behavioral data, we can compute similarity metrics, and here we're looking at some multidimensional scaling again. On the left and right, we actually have the exact same plot. I just like looking at symbols, but it occurs to me now that it's a bit faint for you to see at the back, so I'll draw some circles over here just to give you the summary, really. What these results tell us is that human faces are perceived as visually distinct from all other objects, which is what we might expect. But what's interesting is that pareidolia, illusory faces, is spontaneously forming its own category, separate from human faces and also separate from matched objects. So participants are sensitive to this illusion, even when they're not cued to look for faces or not asked anything about the faces in these images. The end goal now is to try and map this pattern of perceptual similarity to brain activity using EEG recordings, and I think we're 20 subjects into that experiment right now, so watch this space, I guess. So I quickly want to touch on causal experiments that I did with macaques. Typically at this junction I will talk about the work that I did with amygdala lesions and tying the perception of face pareidolia to the amygdala. But those studies were sort of published in 2018, and instead I wanted to show you some new data. In this particular experiment we were more interested in the contribution of the cortical face patches. And the motivation from this experiment actually came from the fMRI data, where we first saw that the lateral face patches are engaged more by illusory faces than matched objects. And then this was quickly followed up with the electrophysiological experiments. So mostly we targeted the face patch known as ML, the middle face patch. And the neurons in this region, they respond nicely to faces more so than objects, but they also respond more to pareidolia than objects as well. And the relationship between the way they respond to faces and pareidolia is nice and strong and positive, as you can see there. So this is a neural correlate that would normally tell us that this area is somehow engaged in processing faces and also in illusory faces, examples of face pareidolia. So, should go over a few more details here. So we plan to silence these neurons in the ML face patch unilaterally and measure viewing preferences towards the stimuli presented in the contralateral hemifield. So what I'm showing you here is we labeled the mucimol. Mucimol is a GABA agonist. It silences activity for about a 24-hour period in any cortical region. We labeled it with gadolinium in order to confirm what part of the brain we were actually targeting when we infused mucimol. We did try manganese for anyone that's interested in contrast agents, but the time course is extremely difficult to deal with. I didn't realize this from my sort of very basic readings that I did. Manganese jumps synapses in a period of time that varies between 3 hours, 6 hours, 12 hours, and 36 hours. So really good for tracing networks, really bad for this purpose, which is just basically marking where it is that we're infusing our GABA agonist. So here I'm showing you a gadolinium scan. Gadolinium shows up in a T1 weighted MRI scan as just a bright white hypersignal within about half an hour of the infusion. So that's what you're seeing up the top there. You can see this kind of, I don't know if I can point to it here. Oh wow, I can. So that's a little bright white hypersignal there, just kind of telling us where it is that we were targeting with our mucimol. Down the bottom is the same coronal slice, just superimposing the functional localizer data to confirm that we did hit the ML face selective region. now I'm just flexing a little bit. I'm showing you more slices from the same scan just to show you just how precise this particular method was. We're very, very proud of our targeting protocol, so everybody just sit there and, I mean, sometimes it's the little things, right? So awesome stuff. The nice thing about mucimol is it does form what we would call a reversible lesion. So the effects of mucimol are metabolized within 24 hours, which means what we could do is target face-selective cortex in an animal on one day, let them rest for one day, and then we could go back and target an object-selective region on the second day, same animal. Very powerful technique. In any case, we're now looking at the behavioral data, which is essentially looking time at those contralateral images. And what we find is a preference for contralateral real faces that remained regardless of whether we were targeting the object or selective region. So they always looked more at faces than they did at objects. The preference for contralateral illusory faces, on the other hand, disappeared relative to matched objects when the face selective region was silenced, and was enhanced relative to matched objects when the object selective region was silenced. So our general conclusion at this point is that when face selective cortex is silenced, the perception is dominated by the object identity, and when the object selective cortex is silenced, the perception was more dominated by the face identity. This is all just data from the one animal. We do have data from a second animal that was very consistent with what you're seeing here. This suggests yet again that examples of face pareidolia have two identities. One is an object, the other is a face. And I think an interesting challenge to kind of think about as we move forward is how to show whether, or how to test whether the neural representation of face pareidolia is somehow in between faces and objects. So they're kind of a bad example of a face and perhaps a confusing example of an object, or whether when we show examples of face pareidolia, what we're seeing is activation of two selective systems in the ventral visual cortex, the face selective system and the object selective system, and that sort of ends up in some kind of bottleneck later on in visual processing. I think all of this will be really interesting and important to understand in terms of us improving our understanding of the functional organization and operationalization of the ventral visual cortex. So this journey has to begin with a correction of an asymmetry that is crept into my studies of face pareidolia, but also the other studies of pareidolia that other people have going on in their labs. And that is this tendency to overemphasize the face. I think I sort of pointed at this with some of my other tasks, but also there are tasks that other people are doing, where they're basically asking people to perform face non-face tasks, or always asking about the face. There are issues with cueing, priming, also issues with decisional criteria. Also, we never ask about the object's identity. So if it's not a face, then we just kind of say it must be not a face or just an object. But faces are also an object. So we sort of have a very poor sort of understanding and characterization of the object's actual identity, which is actually the ground truth of the situation. So some students, cleverer than myself in my lab, are now coming up with these dual tasks where they show illusory stimuli in face detection tasks, which is the same thing we've always been doing, but also in food or object detection tasks. And of course, food is a really good category. It's a really nice category of biologically relevant stimuli, just like faces, highly variable in terms of their textual properties and all the rest of it. And also pareidolia shows up a lot in food. So that is very helpful. And what we generally find is that pareidolia never performs as well as real faces in face detection tasks, which is what we've seen before. But we find advantages for food items with illusory faces. So this is kind of reinforcing this idea that the perception of those illusory faces is involuntary and spontaneous in some way. All of that said, I know that not all of the answers are going to come from face pareidolia. I know it's just a cute little party trick, but we have lots of other lines of investigation in the lab looking at other kinds of stimulus categories. For example, a parallel line of investigation is using animal faces to investigate whether perceptual similarity or social attachment predicts activation of the face selective regions in the human brain. This idea is based on the observation that in the real world, while monkeys may look more similar to humans, a lot of people have a very strong social bond with their pets, including dogs and cats. And so we're interested in whether these face selective areas are responding to these social agents, that social connection between creatures, or whether it's something to do with just how similar faces are to human faces. And I'm also interested in this idea of faces versus whole agents. So one evil that face people tend to do is lob heads off bodies, right? And then we present bodies in different experiments and we say, hey, look, we've got body selective and face selective neurons. But to some extent, that logic may also be fairly circular. So this gull horse was an image that Brendan Ritchie and I used in a recent review where we went over all the evidence for the independence of the face selective and body selective networks. And we basically just wanted to show people that these things are really tightly connected. Even when they're not meant to be integrated, they tend to be integrated. And so we're really interested in what happens when we add bodies to faces, or for bodies, what happens when we add heads. So sadly, that's all I want to try and get through at least this second question. So normally I'd pause for a little bit of contemplation and questions, but if there's anything about face pareidolia, we'll ask it at the end, because I'm sure I'm running out of time. I'm 100% sure I am. So I'll try and quickly get through this second part. How do we recognize different facial behaviors and emotional expressions? So facial expressions are thought to be part of a very simple communication loop whereby a signaler experiences some kind of physiological or emotional state like hunger. I'm sure you can sort of relate to this at this point in time. These emotions, these internal states, contract the muscles in the face, causing a change in facial behavior. This expression is then recognized by the people around you, receivers, who then change their behavior in some way, and this alters the state of the signaler. So this is kind of meant to be how we reinforce the connection between facial expressions and psychological and emotional states. As adults, there are meant to be at least 7 discrete emotional states that we can reliably signal to receivers. These expressions are thought to be universal and hardwired because they stem from emotional states that are shared by all human beings and other primates. And it follows that expression recognition on the receiving end of this loop would be governed by specialized mechanisms tuned to the appearance of specific facial expressions. This is a really nice theory. None of it is right. So we now know that emotional labels are not universal. There is no concrete link between internal states and muscle contractions. People can't recognize the difference between positive and negative states when they're given naturalistic stimuli. But when you show them staged or posed visual expressions like the ones that I'm showing you here, what they can recognize is that these are fake. These are really disingenuine and staged. So where does that leave us when we're interested in the neural mechanisms processing all of these facial expressions and responsible for recognizing these facial expressions? Well, it means we basically have to go back to first principles and think about the stimuli that we're using in these tasks. And so when I got to NIH, I felt the question that really needed to be asked was actually quite simple. Do our current models of facial expression recognition hold up under naturalistic demands? Now, I was working with macaques and primarily fMRI at the time, so really this was an fMRI experiment that was just designed to verify that naturalistic photographs of facial expressions activate the green face patches known in the literature as MF and AF. MF and AF are typically positioned in the fundus of the superior temporal sulcus. You see them here in this individual's individual localizer data. There are some labels there for you. We reasoned that these patches would be more activated by emotional facial expressions than neutral facial expressions in an fMRI experiment. That was really as much of a verification as we wanted. Luckily, I have a very large collection of photographs taken of macaques in the wild, but the challenge was how do we select the right images to represent emotional and neutral conditions. The conventional approach is to use specific combinations of visual features to code stereotypical facial expressions. For example, according to the macaque coding system, the combination of action units 12, 16, 25, and 26 is the silent bared teeth display, or CBT for short. Cognitive neuroscience, we've always called this expression the fear grin or the fear grimace. Now, in previous research, faces with fear grins had been shown to elicit stronger responses from macaque temporal cortex than faces without fear grins. So it seemed to us that using the macaque coding system to select photographs of macaques in the wild with and without so-called fear grins would be the strongest test of whether our current neural model of face processing holds up under these more naturalistic circumstances. So here we have some illustrative examples of the faces selected to represent these two conditions. Even though I assume none of you are certified macaque coders, I think it's really easy to see how the faces in the red fear grin present condition differ from the faces in the blue fear grin absent condition. However, to acknowledge some of the stuff that I was talking about before and this argument that maybe fear grins don't always signal fear per se, we created a parallel stimulus set, selecting stimuli based on context, not on specific action units or facial features. So to do this, my postback at the time, Amanda Patterson, found videos of macaques on YouTube. I didn't know that was a thing, but it definitely is. And she selected videos where macaques were scared, intimidated, startled, or frightened by something in their environment. and then she took a single frame from those videos to represent that response. The more neutral faces were selected from videos where macaques were just going about their regular monkey business. I will note that because of the nature of this selection process, these images were much more degraded by circumstantial factors than we usually allow in face perception studies. Circumstantial factors like bad lighting, motion blur, head turns, they're much more degraded than what I showed you before in the feature-based set. Needless to say, I was extremely pessimistic about how a contrast comparing these two conditions would work because I believed there was just too much variation in these faces to find anything. And this is just one example of, I'm usually wrong. But then that's a bit of a spoiler alert. Moving on. Finally, we also included a set of human faces in this study. These are photographs taken of actors that were instructed to pull fearful or neutral expressions. It's the gold standard in the human processing literature. In this context, though, these stimuli provide an important contrast because they address the concern that when we're comparing fearful and neutral faces, what we're really doing is comparing faces that differ in terms of low-level visual attributes. For example, fearful faces tend to have local regions of increased contrast because their mouths are open, their teeth are showing, their eyes are wider, and these create these kind of changes in the contrast envelope. If these kinds of differences in low-level visual attributes are driving valence effects, then these stimuli should be just as effective as the macaque faces that I showed you before. So in this study, we scanned 3 subjects in a 4.7 Tesla Bruker scanner, and we also used a contrast agent enhanced fMRI technique. I don't know that I mentioned that before. This just means that we inject myelin before a scan session. And it also means that what we're measuring is much more closely related to cerebral blood volume than the bold signal. The subjects, again, were awake and fixating while we presented them with blocks of faces that varied in emotional valence as well as stimulus selection method. This is what the scanner looks like when you turn it vertical. People are always kind of fascinated by this. It looks a lot more like a rocket ship than it does an MRI bore. And we sort of raise the monkeys up like we're offering something to the scientific gods every morning. It's a lot of fun. So turning to the results, here I'm going to plot average valence effects such that if the response is above 0, that would indicate that fearful faces drove a greater response than neutral faces, as we're expecting. If the response is below 0, that would indicate neutral faces drove a greater response. And I'm going to show you these valence effects across the 3 sets of stimuli, selected based on context, MacFacts, and human behavior. This is to remind me to tell you that I'm plotting the percent signal change for all voxels in the ventral visual cortex, anatomically defined, not the face selective network. So surprisingly, what we found is that the stimuli selected based on context drove the largest valence effects. The bars here are the results averaging across subjects. There's only 3 of them. So I'm also showing you 3 red lines representing the individual subject data. Typically in fMRI experiments with non-human primates, what we use is a fixed effects approach. So when we take that approach, that is what the data look like. But all of these results indicate that the faces that were selected based on context did a better job of evoking valence effects than those based on MacFacts or features or human behavior. Now I'm just showing you the contrast projected onto the inflated cortical surfaces as before for the 3 stimulus conditions, and I'm going to zoom in on the results for the context-based condition because that's the only time that we see anything when we're averaging across subjects. And here what we can see is that the activity is again not clustering in the fundus of the superior temporal sulcus, but rather these yellow hotspots of activity are positioned in the lateral convexity of the brain in area TE and TEO. So we're not the first to find this. In fact, when you dig hard enough, all the studies that have measured fMRI activity in macaques and have compared the response to emotional faces and neutral faces have reported pockets of differential activity in the lateral convexity in area TE. Previous data from Leslie's group had revealed that there's no relationship between face selectivity in the ventral visual cortex and valence effects for face stimuli, and we find that also here. So all of these results are marrying really well to previous reports, just not really well to the theories that we have describing facial expression recognition. So going back to this map, if I now draw a green outline to delineate the face selective network, you can clearly see that the yellow regions that are activated more by fearful faces are falling outside of that conventional network, again, more lateral in area TE. So in sum, these results demonstrate that when we use naturalistic facial expressions, we find that the ventral visual cortex is sensitive to differences in facial signals, but these effects do not emerge where we expect them to. And in my view, one of the most interesting findings in these results is just how ineffective the feature-based condition was. I mean, I just don't understand why these faces, with all of their variance and kind of overlap, I see different facial expressions in both of those groups, would necessarily elicit a stronger valence effect than these faces where we can see with our eye that there is a particular difference between the faces on the left and the faces on the right. So to answer this question, we need to turn to macaque behavior, because obviously we have very bad examples of macaques. We need to ask them why these faces are working better than others. And I'm going to try and really quickly get through some behavioral data here. So these macaques, 4 macaques were trained to perform a condition 2 alternative force choice task. During training, the subjects were rewarded for selecting the most pleasant face, which are, in this context, neutral faces or lip smacks, kind of like smiles to them. They look a little bit like kisses or something. We had noticed in a previous experiment that these subjects were avoiding faces that were threatening them, that looked aggressive. So we were just trying to lean in and reinforce the spontaneous behavior that these subjects already had. Once they reached a certain training criteria, we probed these subjects with trials of depicting pairs of faces that they had not seen during training. These trials were not differentially reinforced. We were just looking for an indication as to which was the most pleasant face. The results averaging across subjects indicate that unlike lip smacks, positive faces, or open mouth threats, negative faces, which are in red and purple respectively here, fear grins varied in how frequently they were selected as the most pleasant face. In other words, not all fear grins are unpleasant signals conveying fear. Instead, their meaning seems to vary. And to better illustrate this point with facial signals we can actually understand as humans, let's think about smiles. In terms of action units and exact facial features, both of these faces qualify as happy faces, but their meaning to us is entirely different. The one at the top is signaling affiliation and increased approachability, whereas in the one down the bottom is triggering the uncanny valley and is signaling some kind of threat. So going back to the fMRI data, the observation that fear grins alone did not drive strong valence effects may not be that surprising, because perhaps what we were doing was averaging across the response to different kinds of fear grins, some pleasant and some unpleasant. And if that's the case, the bigger picture here is that it seems possible that the ventral visual cortex is encoding socio-emotional meaning rather than putative visual properties. I've got another example here from bodies, but I'll leave that for now. All of this to basically say that, again, quite a few times with different observations, we've made this argument that perhaps visual representations of high-level visual stimuli are not organized according to perceptual similarity, but something else. And that something else may be socio-emotional meaning. But it is very hard to build this argument in the macaque world because we are not macaques. So if we want to argue that something is in the stimuli that isn't just basic image properties, we really need to port this line of investigation back into the human realm where we can do some more detailed behavioral experiments. To this end, 2 students in my lab have collected a large set of 1,000 naturalistic facial expressions. These images were largely sourced from YouTube, and we refer to them collectively and lovingly as the WFD, or the Wild Faces Database. But instead of just finding a set of faces and assuming we're capturing naturalistic expressions that everybody can read and recognize, we're taking a data-driven approach. So in an online experiment, we asked over 200 participants to rate these faces for emotion on a 9-point scale, which included other and unknown. And we asked them to indicate how intense and authentic each expression was on a sliding scale. So for the sake of comparison, we also asked participants to rate 1,000 faces sampled from existing databases to include Ekman faces and NIMSTIM faces, and 1,000 faces randomly selected from the 10K US Adult Faces database, which is a database of face images scraped from the internet without any further selection biases. So using the responses to the expression categorization task, we computed each image's modal score. When we then look at the distribution of these scores, we see that both the wild faces and the existing databases have this nice spread, whereas in the unbiased scrape on the far right there, basically all of those faces were rated as neutral or happy. Here I'm showing you 3 examples of faces that were rated by the majority of participants as happy from the 3 data sets, and I think that kind of is consistent with our phenomenal experience of these faces. Importantly, for every image, we were also able to compute a score to reflect consistency across participants. So for example, if an image was always rated by happy by all participants, then the score would be 1. But if a face was rated by happy by some participants, but surprised as other faces, its score would be 2. So on and so forth. So on this scale, 9 would be the highest possible score because that would mean participants used all 9 possible emotional categories to rate the image. When we look at the distribution of these scores, we see that the wild faces had a higher median score than the other databases, which means that there tended to be less agreement across participants when they were rating the wild faces. So here we have 3 different wild faces, or wild face images, all rated by the majority of participants as happy, but they differ in terms of their consistency. The face on the left was rated by all participants as happy all of the time. The face in the middle was rated as happy by 84% of participants, but other participants rated it as surprised, neutral, or other. The face on the right was rated as happy by 32% of participants, but as many as 6 other emotions were used to categorize that face. So in the past, these kinds of experiments really only took into account the modal score and considered that the most important thing, with all other responses treated as mistakes or noise. But I think all of these responses in these data should be treated as equally valid, and that they actually help us contextualize something that is very real in these images. So here are some examples of angry faces that differ in consistency. I enjoy the fact that this scale seems to go from road rage through to disappointed parent. One thing you might know is that notice is that the middle face is not human. It's an example of face pareidolia, old habits die hard. Also, we included a few animal faces and examples of face pareidolia in the WFD to provide as much variation in these images as possible. An interesting subcategory of these faces is those that were most frequently categorized as other. Participants were given the option to say unknown, so by selecting other, they were indicating that they knew the emotion being signaled, but it was not one of the other options. In my view, a lot of these faces look confused or in pain, but maybe that's just because those are emotions I identify with, I'm not sure. I don't have any information of what the actual emotion is, but these are the examples of the other faces. So we also got information about intensity and genuineness, and in terms of the WFD and the unbiased scrape, the more intense the emotion was, the more genuine it was perceived as being. The relationship was actually negative in the existing databases, so the more extreme stage facial expressions tend to be, the more disingenuine, or they lack some kind of genuineness at the top end of that scale. We also looked at some multivariate approaches here, and we're moving on and looking at even more detailed behavior where we're not cueing people about emotion. So this is again using that triplet oddball task that I suggested, that I talked about a little bit at the beginning of the talk. So far we've collected 580 participants, so over 232,000 individual trials using these faces. There are a lot of stimuli, so to understand perceptual dissimilarity, we have to run a lot of trials. We're also doing this for the NIMSTIM database, and I feel kind of bad about that. I don't mean to pick on people as having a particular kind of or using a particular kind of stimulus. Really, I just want to compare naturalistic faces to these more staged and posed faces, and that's a really great database to use. So all of this is going to help us. So this is not real data. I'm sort of generated this data, or it came from another figure of some description. But we're going to be able to describe the perceptual dissimilarity between the faces in the WFD. And from there, we can do model comparisons to figure out how people are actually responding to this task, as well as some eye tracking and some EEG. And finally, the WFD is freely available for everyone to use, as is the style these days. It's 1,000 faces. All of the behavioral data is there. You need not ask us for it. It's all freely available. So if anyone's interested in doing experiments with naturalistic facial expressions, I would encourage you to go for it. And as I suspected, I do not have time to talk about context dependency. So I'll just give you a tantalizing summary slide. So instead of just looking at emotional valence, which is something that we look at a lot in the lab, what I wanted to do was start using video stimuli. And so we had these naturalistic videos that varied in valence, but also something that I used to call social distance, but now call context. And the primary difference between this manipulation of context is that some of these interactions, the social agent is looking at the camera and therefore at the subject when they're looking at these videos. We call this interaction mode. And sometimes the subjects are just watching like reality TV. They're watching third parties just kind of interact with each other. And what we are finding, just using these video stimuli in an fMRI experiment, is essentially the connectivity, the functional connectivity within the local face selective network is a lot stronger. We see a lot more activation of the face selective network when social agents are interacting with the subject. When we're just watching third-party interactions, there seems to be a lot more connectivity within the body selective network and a lot more connectivity to the motion-sensitive areas of the brain like MT. So that's just a little bit of an upshot there. Oh, look, this is just a summary slide of everything I went over today. I am sorry it was such a gallop. I didn't want to keep you for a long period of time, but here we are. I should end by thanking all the various people that worked with me, that I sort of drag into these experiments from around the world, and of course my core funding sources who have to track everything I do. So thank you.
Wow, thank you so much.
That's a lot of stuff. I apologize.
I was like, oh, what's next? It's just amazing. And do you sleep much?